Hello, my name is Lukas Zygonek. I'm a group leader at the stem cell unit in Göttingen, and I'm happy to present you our recent data with the title Custom-Tailored Cardiomyocytes, the Directed Differentiation of Human iPS Cells into Defined Atrial and Ventricular Cardiomyocyte Subtypes. Just to give you a short introduction of our unit. So we are a technology platform at the University Medical Center in Göttingen, and our services are mainly focusing on reprogramming of patient-specific iPS cells, in genome editing of these iPS cells, in the differentiation of iPS cells mainly into functional cardiomyocytes, and also we have a collection of iPS cell lines of right now more than 120 patients. In total also 400 then well-characterized iPS cell lines, which mainly focus on cardiac diseases. And we are very happy to share our expertise and also our resources from the iPS cell bank with the research field. So just feel free to check our website and contact me. Besides the service, we are also focusing on research a little bit, and the main focus is the development and the implementation of new methods and technologies for the directed differentiation and the genome editing of iPS cell lines. And in more detail, I'm also interested in disease modeling of cardiovascular diseases, in drug screening, and also in tissue engineering. So when we now talk about cardiovascular research, cardiovascular diseases are the principal cause of death in the world, in Europe with over 4 million deaths per year. And also important to know is that cardiovascular diseases are the very important cause of healthcare expenditure in the world. So not just for us, but for all of us, it is of great clinical importance to understand the molecular and functional disease mechanisms that lead to cardiovascular diseases, also to enhance the diagnostic tools, and to develop at the end new therapeutic strategies to improve the cardiac function. So also when we take a closer look into the drug discovery, we know that we start with around 10,000 compounds that are screened and end up at the end with just one substance that is launched. That means we have huge costs and a low overall success from target to launch. And reasons for this are a lack of selectivity. We have limitations in safety, efficacy, and also cardiovascular toxicity. And one major cause is based in the preclinical trials. So therefore, we utilize non-cardiac human cell lines for in vitro assays or animal cell models. And these models not accurately represent the human cardiomyocyte physiology and also therefore have limitations in predicting drug responses on the human heart. Therefore, we have a huge demand for human, more native-like models that really can recapitulate the complex biology of the heart for a better translation and for better safety. And we and all of us hopefully believe that iPS cell technology have a huge opportunity for disease modeling, for drug development, and also for toxicity screenings. We ourselves are mainly interested in cardiovascular diseases. What we have is, so we collect samples from patients with genetically caused cardiovascular diseases, and we create patient-specific iPS cell lines. Furthermore, what we also can do, we use CRISPR-Cas technology to generate mutated isogenic iPS cell lines. This iPS cell lines can then further be differentiated in a certain cell type of interest, like for us, for example, iPS-derived cardiomyocytes. And these cells can then be used for various applications, like to understanding the disease, for drug screenings, for toxicity screenings, as well as for regenerative applications, by transplanting cells or even tissues back to the patient. And all these approaches, we really hope to help the patient at the end. When we now take a closer look in the cardiogenesis, so iPS cell lines can really differentiate into cardiomyocytes, and various signaling pathways that are important during this whole process are already known. And the idea in our cell culture is to influence the signaling cascades with small molecules to really induce the cardiogenesis in vitro. And the commonly used protocols, which are, for example, the ones from Leon et al. And Burrich et al., really provide a very high yield and a high purity of cardiomyocytes. However, they always end up with a heterogeneous population of ventricular, atrial, and pacemaker cell-like cardiomyocytes. Most of the cells are ventricular cardiomyocytes. We have some atrial and a few nodal-like cardiomyocytes. And this heterogeneous population really have then strong limitations in their final field of application. And when I'm talking about cardiomyocyte subtypes, dependent on their localization and their requirements, they totally differ in their molecular and cell biological as well as in their functional properties. When we now take a closer look to the electrophysiology, dependent on the cell type, they really show different action potential morphology, which is then of course caused by expression of different ion channels. So we have some ion channels that are typically expressed in equal levels between ventricular and atrial cardiomyocytes. However, we also have some ion channels that are only expressed in atrial cardiomyocytes, leading then to the different action potential shape. And we and others believe that homogeneous population of cardiomyocytes could more precisely recapitulate the cardiac disease, since many cardiac diseases are often subtype restricted. And we believe that homogeneous populations of certain subtypes could more precisely predict the pharmacological effects in drug development and also in toxicity screenings. So the goals of our study were to, first of all, develop an efficient and cost-effective method for the directed differentiation of iPS cells into either ventricular or atrial cardiomyocytes in feeder-free culture conditions and monolayer-based. And finally, what we also wanted to achieve is to obtain a more comprehensive understanding of the iPS-derived cardiomyocyte subtypes, which is really required if we want to go in all our different fields. And our protocols are based on the Wnt signaling, and for the atrial differentiation, we choose retinoic acid additionally, and I will tell you why. So retinoic acid is implicated in various steps of cardiovascular development and also in atrial chamber development. This is known from various animal studies in the previous years. And while the H2, which is the main enzyme responsible for the synthesis, is expressed in the posterior mesoderm, leading to establish a boundary of the second heart field, shown in green here. And when we have mutants of YLDH2, we have an expanded second heart field. So we know retinoic acid seems to play a role in the second heart field, especially since it is known that the second heart field also contributes to the atrium. Furthermore, retinoic acid is important for the expression of Hox genes, and we know that different Hox genes, especially Hox B1, A1, and A3, the downstream targets of retinoic acid, are expressed in the atrium and in the outflow tract. Therefore, we have chosen retinoic acid in our cell culture to test it if it really can differentiate into atrial cardiomyocytes. So, we were able to establish a protocol that mainly yields in pure ventricular or atrial cardiomyocytes, and this is based on the temporal modulation of canonical wind and retinoic acid signaling. We really evaluated the time window and the concentration of retinoic acid and came up with this comic. So our cells are typically cultured in essential 8 medium, and when we start our differentiation, we start with wind activation by a cheer for 2 days. Then we perform a wind inactivation by IWP2, and then we have the cardiac induction done, and we just wait till the first contracting cardiomyocytes start to beat around day 8, and further cultivate the cells up to day 60 for further analysis. When we wanted to achieve pure atrial cardiomyocytes, we added retinoic acid at a concentration of 1 micromolar between day 3 and day 6 into this culture and came up with nearly only atrial cardiomyocytes. So we were able to show that wind signaling is not really different between ventricular and atrial cardiomyocytes. However, when we take a look at the retinoic acid signaling, we see that the signaling is highly increased, and the genes upstream, the downstream targets of retinoic acid are really upregulated in the atrial differentiation. When we then take a look at typical cardiac markers, we see that general cardiac markers are equally expressed between both subtypes, especially also at day 60. However, the ventricular markers are highly expressed in the ventricular iPS cardiomyocytes. However, they are really absent in the atrial differentiation, and it's the other way around when we take a look at the atrial markers, which are around day 60, not really highly present in the ventricular differentiation, but are really upregulated in the atrial differentiation. When we then take a look at the yield, we really could not see a significant difference between both differentiations and ended up with up to 99% pure cardiomyocytes, or for ventricular ones, and 95% for atrial cardiomyocytes. So with this, we are really convinced that we established a very efficient protocol for the differentiation into the certain atrial or ventricular subtypes. Then we take a look at the morphology and of the cardiomyocytes. So we performed immunostainings of different cardiac markers, and strikingly, we were able to see that the ventricular marker MLC2V is highly expressed in the ventricular differentiation. However, it is nearly absent in the atrial differentiation, and it's the other way around when we take a look at the atrial marker, which is only little present in the ventricular differentiation, but really highly upregulated in the atrial differentiation. Then we take a closer look at the cardiomyocytes, and we have seen that both subtypes display a well-organized striatic pattern, visualized by alpha-actinine. We have gap junctions, which are not very clearly seen here, but in green. So we have gap junctions labeled by connexine 43, and we have striated patterns of the sarcomeric reticulum marker ryanodine receptor 2, which is the main calcium release unit in cardiomyocytes. And we were able to confirm our results also in Western blot analysis. Again, we see that MLC2V is highly upregulated in the ventricular differentiation, but totally absent in the atrial differentiation, and the other way around for the atrial markers, which is expressed at low levels in the ventricular differentiation, but at very high levels in the atrial differentiation. We also performed a quantification of our results, and we could see that around 88% of the ventricular differentiations express also the V marker, but only 9% express the MLC2A marker, and it's the other way around for the atrial differentiation. So with this, we can see that we efficiently can generate nearly homogeneous populations of either atrial or ventricular cardiomyocytes. We went further and performed RNA sequencing, and we could see that the samples cluster dependent on their differentiation. So we have the ventricular differentiation independent of the cell lines that were used, clustered together, and the atrial differentiation also clustered together. With this, we were able to identify more than 3,000 differentially expressed genes, including, of course, typical cardiac markers, which are expressed at equal levels. We were able to identify genes that are really upregulated in the ventricular differentiation with many known markers like MLC2V, for example. And it's the other way around for typical atrial markers that are really upregulated in the atrial differentiation. And strikingly, we were able to see that also the ion channel expression showed a strong correlation with the different subtypes. So as I said before, we have some ion channels that are expressed at equal levels, labeled here in black. You see that they are also expressed at equal levels between the different differentiations. We have some genes like KCN J2 and J4. They have a higher current in ventricular cardiomyocytes. Therefore, we also have a higher upregulation of these genes in the ventricular differentiation, and it's the other way around for the atrial current, which are only present in atrial cardiomyocytes, and also the genes are very upregulated in the atrial differentiation. This is the RNA-seq data, and we went further and wanted also to perform proteomic analysis, since SILAC, in combination with the quantitative mass spectrometry, is believed to be a powerful tool to investigate the proteome at very high resolution. However, SILAC was really limited to dividing cells. We were able to establish SILAC labeling also in non-dividing iPS cardiomyocytes by prolonged culture up to 45 days, where we were able to label more than 98% of our cultures with heavily labeled SILAC medium. With this, we were able to identify more than 3,500 proteins, few of them being differentially expressed between both differentiations. Again, we could confirm that this protein profiles correlated with the expression of the human ventricle and with the atria, but we were also able to identify some novel markers with potential crucial roles in cardiac subtype specification, or maybe their cellular function, which has to be investigated in more detail in the future. Then, of course, we wanted to perform the functional analysis, which is really important when we talk about cardiac subtypes, and we were able to establish a fluorescence-based high-throughput characterization of the action potentials and calcium transients, which are typically different between both subtypes. And we used di8 ANEPS as a voltage dye to record an optical action potential, and we used fluor4 in the same cell to record the calcium transients. And with this, we were able to visualize the action potential and the calcium transients in a similar cell and to quantify this in more detail. We could show that the action potential duration APD50 at 50% is really shorter in atrial cardiomyocytes compared to ventricular cardiomyocytes. We could show also that the calcium transients, both time to peak as well as the decay, is significantly shorter in atrial cardiomyocytes, which really correlates with published data. Again, we also performed the spontaneous beating frequency, which is also in line with published data that atrial cells contract faster than ventricular cardiomyocytes. And at the end, we do not only wanted to analyze single cells or cells in 2D, but also wanted to create atrial and ventricular tissue. And we collaborated with colleagues from Göttingen, who are really experts in generating engineered heart muscles, which is a mixture of atrial or ventricular iPS-derived cardiomyocytes, around 70%. They are mixed with 30% fibroblasts, combined in a collagen matrix, generating so-called engineered heart muscles. And this force-generating, more native-like tissue you can see here in both videos. You can already see that the atrial tissue contracts highly faster than the ventricular one. And with this, both tissues, we could perform the functional analysis in more detail. So, first of all, we also confirmed the expression of the typical cardiac markers. So we performed cross-sections and stained for the MLC2V for ventricular markers, which is highly upregulated in the ventricular tissue, and the atrial markers, MLC2A, is only present in the atrial AHMs. And when we look at the contraction curves, we see that the contraction curves here in red of atrial cells are significantly shorter, both the contraction as well as the relaxation time, and the force, surprisingly and interestingly, was not significantly different between both subtype tissues, but we were able to see a positive force frequency effect in ventricular tissues, but not in atrial tissues, which is in line with already published data from human atrium and ventricle. We also performed pharmacological analysis, what I'm not showing here right now, and we could see that the atrial tissue behaves different to atrial selective drug carbachiol, whereas we have not seen a huge effect of this in ventricular tissue. In summary, I hope I could convince you that we established a protocol for the differentiation into atrial or ventricular cardiomyocytes by temporal modulation of the canonical Wnt and retinoic acid signaling, which really results in nearly homogeneous populations of either atrial or ventricular cardiomyocytes. We performed transcriptome and proteome profiling of both subtypes, which really highly correspond to human atrium and ventricle, and the functional analysis, which we performed by optical action potential measurements and calcium imaging, as well as recently shown by engineered heart muscles, really demonstrates that they really correlate to human atrium and ventricle. And with this, we really hope to get a better understanding of the molecular and functional profiles of both subtypes, and to get a better suitability of these cells in tissues for further applications in disease modeling, drug screening, as well as cell-based therapeutic approaches. And with this, I wanted to thank all our collaboration partners and my group here. And if you have further comments, questions, requests, or collaborations, please visit our website and contact me. Thank you very much.